Thank you. My question is for the first speaker. Could you expand a little bit more on the differences between the brain response to the single pulse and to the clinical used multiple pulses in terms of the scientific investigation?
Sure, absolutely. So I think the bursts of the RTMS, it's the only way that I've seen where you get really good site of stimulation bold, and it may be necessary to have something like the Vienna coil or to do the bursts, one or the other. So I don't get good site of stimulation, and there was some discussion of that before. What I do get is a nice evoked response in some of these subcortical regions. So if what you care about is a mapping and a circuit integrity perspective that you can look at ahead of time to potentially inform the clinical outcome, the pings for us seem to work very well. But the RTMS, again, it's not exactly what they're doing in the clinic because you're getting a much, much smaller dose. It just happens to be that the burst pattern coincides with what you're doing in one of your sessions in the treatment. So there's still, no matter what we're doing in the scanner, there's a discrepancy between what we're doing in the clinic and what we're doing in the scanner. So it's important for us to validate what we're doing in the scanner with the actual clinical outcome to convince you that, hey, the way we're pinging it or bursting or whatever is actually telling you something about that person's response to TMS and put it in the context of something clinical.
Great. Thank you. Yes, one more.
Yeah, thanks for great talks. I have a question also to the first speaker. When you explained the pinging, could you elaborate a bit on if you think you're actually picking a subgenal status of the subject? So are you actually probing, say, the degree of dysfunction, or are you actually just technically pre-testing the success of your stimulation? I didn't quite get the concept.
Yeah, so I think I can only guess because we don't do longitudinal assessments without doing an intervention. So it could be a state-dependent thing. The way we think of it is like people would hope that DTI sort of informs circuit hypotheses. So you're like, there's a connection between A and B in this person, and it's pretty strong, and not necessarily capturing like what happens when their mood is worse versus what happens when it's better, and does that relate to something? I think there's a rationale for exploring those state-dependent and illness symptom kind of dependent effects, but I think in our data, that's not the way we're going. I'll also say that resting connectivity hasn't been a great predictor for us. We collected a baseline. I'm kind of curious about other people, like how long they go between their baseline connectivity and when they actually do the treatment. In our data, it looks like it matters. We take a worse sample closer to the treatment, and it is a predictor. Put it in the same model with the ping. Only the ping survives, so it explains the connectivity. So I don't have huge confidence in the connectivity value being really clinically meaningful. I think other people have papers suggesting it's problematic. Some people still hold out hope that we could clean it up and make it better. But for me, the ping is just so straightforward interpretation-wise. It's like engagement of the circuit, stimulate A, measure B, and it seems to pan out for us.
Yep. Just a really quick question for Katie. In terms of the clustering, have you ever considered looking at a model that would model the data potentially more parsimoniously as a continuum or as a dimension? Yeah. I think sometimes with clusters, at least in my own experience, I think you use the CH metric, you had a suggestion of 4 clusters, and then you use something like the silhouette or the gap statistic, and it might take 2 or 4. And so all those solutions are stable. So if you then test that in another data set, the 2, 3, 4, or perhaps 5 are stable solutions. But that might mean that a more continuous or a dimensional approach provides a parsimonious representation of the variation across your CCA modes. And so is that something that's worthy of considering?
Yeah, I mean, I think I'm definitely in the favor of a more continuous solution. I think we are probably washing out meaningful signal by subtyping individuals. And I agree that if you look at silhouette versus gap versus CH, like you may get slightly different results, but significant ones. So I'm definitely on the continuous camp, not the categorical one. But I understand the clinical merit of decision-making, clinical decision-making with subtypes. And so for the purposes of this paper, we really just wanted to show that these are still stable. So if you really wanted to proceed that way, it is still okay. But do I ascribe more one way or another? Yeah, definitely more on the continuous camp than the categorical one.
And I guess the advantage of a subtype is at the end of the day, you need a yes or no, right? Either true or you.
Exactly. Yeah, there's no in between for sure, and I think that's where the clinical needs don't necessarily overlap with the ones that explain variability or from a more basic science mechanistic perspective, there are some competing interests with these kinds of solutions for sure. So yeah.
I have a question for you, Katie. So in terms of the DLPFC, where stimulation happened with TMS, the symptoms that responded to that, do those things overlap? And like when you stimulated DLPFC, and if that was part of the whatever is a symptom cluster, does that mean that the abnormality looks like it's reversing and that symptom is also following suit, or does it look more like you shouldn't chase the abnormality, you should chase more intact normal circuits and then the patients respond better?
So that's a great question. What we found was that there really wasn't a difference based on where you stimulated and which subtype or continuous dimension was related to improvement. It didn't matter if you got dorsomedial, left dorsolateral, 10 hertz, or theta burst. It was that anhedonia dimension that was predictive of response. I'm still interested in looking at the dorsomedial, dorsolateral symptom differences, but maybe baseline heterogeneity alone isn't going to be just suited to getting at that question, which is where alternatives might come into play. Where I showed, instead of just looking at baseline symptom severity, you can look at changes in symptom severity. So if you have a training set of individuals and you already know how they got better on different symptoms, you could leverage that symptom improvement data with your baseline imaging and then use that to train your model and see whether or not then the subtypes of response are enriched one way or another for a particular TMS target, which would be a great thing to test, but we haven't done that. We only did that in the SSRI data simply because at the time, this was a few years ago when we started that, but at the time we were kind of interested in it because of the observation that we saw in the MADRS and also the fact that SSRIs are often prescribed not just for depression, but also things like chronic pain, anxiety, and other things. So we really wanted to get at least a first-line intervention and see what was going on. And we actually, interestingly, this is not what you were asking, but the subtype SSRI response subtypes and the baseline subtypes do have some overlap, but we're just about to submit that. So stay tuned.
Thanks.
Thanks so much. So now I think we have a session looking at databases and software packages. Is that right, Hamid?
So we have a few minutes. First of all, we communicated with different kind of researchers in the field in terms of the software packages that people can use. I just shared a document in the WhatsApp group that has a list of different software packages and publicly available databases that people can use to combine different sort of imaging modalities and brain stimulation. So that could be a kind of initial resource for those who are interested to start in this field and learn how to basically follow what people have done so far. So that could be one starting point. There was a high school student in the group that he was introducing himself. And is he here or not right now? He was here. So it might be interesting. One high school student from one of the Columbia preparatory schools, they approached us and they have been interested to learn how they might be able to do sort of MRI and computational head models and dose estimation, things like that, in line with brain stimulation interventions that we are doing. And we basically invited them to our team, and now they are, I mean, high school students these days, they can learn many things that you cannot even imagine in terms of the quality of the data analytics that they can do. And now they are doing lots of computational head modeling, kind of differentiating different electrode montages and predicting which montage is going to receive kind of higher level of electricity in this specific area. So they are doing lots of different things. So even you can engage high school students in this sort of data analytic kind of pipelines. And there are definitely even potentials to have them as a part of the community, because I'm pretty confident that in the future we need to have even kind of younger people to get involved in that process, because we definitely need to have lots of collaboration between different disciplines to be able to do new stuff. So that could be a resource for those who are interested to follow. The other thing, as we have discussed, there are an ongoing efforts to develop different checklists, methodological checklists, to be able to improve the quality of the studies in the field. So we discussed about one of them in Nature Protocols. We are already in touch with the editors of Nature Protocols, and they are interested to publish more of these methodological checklists. So if there are teams that are interested to develop these methodological checklists, we will definitely be happy to help them and support them to develop more methodological checklists. It takes huge amount of time and effort to bring all the people together and conclude together, but at the end of the day, they are going to be very helpful for the community. So if you are interested to push those ideas forward, we'd highly kind of recommend it, and we will definitely be happy to support that. I will add a specific paper that we recently published in Neuropsychopharmacology discussing about all the list of imaging checklists that we already have in the field, and potentially the other things that we need to have in the future. So that could also be a resource for those who are interested. So we hope that this will be a community that people are going to work together in the future. And if other speakers are interested to share any other things that basically can encourage others to collaborate, that might be helpful. One other thing that might be helpful is, as you know, that there is a kind of large consortium, it's called Enigma Consortium, bringing people together in the field of kind of different neuroimaging modalities and different disorders. There is a specific Enigma addiction that we work on, but there is a new Enigma working group that they are working on neuromodulation data and brain imaging data. And I was trying to work with them to have a small presentation from what they are doing in our workshop. They ended up not being able to provide kind of further details for us, but that could be another framework for collaboration, that the labs that are doing brain imaging and neuromodulation are interested to share their databases. They might be able to use that sort of framework. And I think Paul Thompson today in the afternoon, he's going to discuss about, mainly about AI, but I can imagine there would be some discussions about Enigma as well in the Talerach talk as well. So if there are others who are interested to share ideas or potentials for collaboration or sharing the databases, even there are teams, one of the kind of the data resources that you have in the list that we provided is a group in Mexico that they ran a really nice trial, and then they published all the databases that they had, the TMS databases, Eduardo from one of the really good universities in Mexico. And I think it's a really good culture if we kind of run a large clinical trial with TMS and fMRI, and you have lots of databases, and you already publish things, and you are happy to share the database with others. That would be something that would be helpful for others in the field to be able to use those databases. Any other thoughts from other speakers in terms of how we might be able to support these collaborations within the community? Or those who are here in the room, if you have any suggestions, any.
For the data to be shared, is it restricted to the clinical data, or can I just share some data from the LD?
Of course, yeah. I mean, any sort of databases that could be explored by other teams, and they can use those sort of databases, they are going to be helpful. Even the things that Teresa was presenting about the type of noise data that you might have inside the scanner. Sometimes even having those types of databases are helpful for you to just compare your own database. Even we have been discussing before in terms of the labs that are doing electrical stimulation inside a scanner, when they start to collect data, we are happy to receive their database, their initial database, and look into the noise of their databases and support them in terms of, okay, what type of noise you already have that you might need to consider if you want to push a larger study with this new database. So there could be even potential collaboration in terms of kind of doing quality control on the databases that other labs are collecting and helping them to just make sure that, I mean, how they can manage the noise. I mean, Teresa was mentioning about the kind of different sort of way of kind of using the mesh panel or the filter boxes or the other. They make significant difference in terms of the level of noise that you have inside the scan, as you have been mentioning, and how we might be able to help other labs to be able to make sure that they are going to collect good quality data.
Just as you mentioned Enigma, I would like to emphasize that beyond the actual neuromodulation group, the activity level of which I don't know about, there's also methods groups. And I think one particular aspect where Enigma could also produce a benefit for all groups who work on neuromodulation is pipeline harmonization. So Enigma is just not approaching, but starting the large-scale fMRI analysis, both task and resting state. So using these pipelines might be very beneficial if you want to either do biotyping or compare your, for example, pre-intervention patterns and so on. So I think these groups are very open, and also the whole concept is open science and very interactive. Sorry, my name is Philip Salmon. I've been working with Enigma for years, also in the methods groups. So join the talk today of Paul Thompson, and I think...
On the WhatsApp group that we already have, can you just share that idea in the WhatsApp group?
Yeah, we'll do that. Sure.
That others can basically use.
Good. Yeah.
Thank you.
Sort of jumping on that, but from an Australian perspective, our team down in Deakin, down in Melbourne, we've created a big data repository for non-invasive brain stimulation data. It is not yet open to the combined imaging. That is the only caveat. We've launched it this year for the first time, and exactly as you were talking about with the noise elements, we're looking to create the largest data repository of non-invasive brain stimulation data across all populations so that we can look into the intra-individual and inter-individual differences with non-invasive brain stimulation techniques. We're also hoping that we've launched it with a couple of protocol documents. You can find us by Googling big NIBS or big NIBS data online. We've launched our protocol document, so we're hoping that that's going to provide the protocol not so much for the pre-processing. We know how to pre-process imaging data, that, BIDS exists. We understand how to store all that data, but we're going to be supporting researchers who are doing NIBS research into the protocols for analysis and group difference analysis within group analysis with our protocols that are published online. So if you've got any NIBS data that you'd be willing to share, or you have the ethics to share, just search us up on Google, Big NIBS. We are literally taking any technique and any data at this point from any population group, and hopefully we'll get there.